turns upon this. As notoriously, he had that vision when he saw a plate of bacteria that had got mould in it. It was due to be discarded. He noticed the pattern that the bacteria are not growing around the mould. That therefore, he thought perhaps something from the mould is causing it. And because it's penicillium, what came from it is obviously called penicillin. But at the time, this didn't attract an awful lot of notice. Arguably, Fleming, despite his brilliant observation and deduction, would have been a footnote in the history of science had it not been for Florey and Shane. Florey is Australian and Shane is German. They both received knighthoods for their work. They were the chemists who isolated penicillin and also carried it forward in trials and in developing these methods for mass production. So they did the first test in mice, then started to test in humans. Interestingly, connecting into Geoffrey's lecture, in terms of quantity of material and sample, their first test on a human was someone who had got a horrific skin infection, which eventually killed him, from, I think, a gardening injury, a thorn or some such. They didn't have an awful lot of penicillin at the time. They hadn't got the mass production going. So they treated him. He started to get better, and then they ran out. And as I say, the poor chap went on and didn't die. They then actually realised that since they would be able to get higher doses per person, they actually switched to using children as their extra experiments because it would take less per person to heal them. But of course, it was safe, it was hugely effective, and this, and of course, there was an awful lot of effort put behind the research to production because this was during the Second World War. First mass recipients of it were people who were with battlefield injuries. This is the structure. It was deduced by Shane. It's a beta-lactam antibiotic, which means that it gets in the way of the proteins involved in synthesizing the bacterial cell wall. And because it does so, then the wall self-destructs. The structure was verified by Dorothy Hodgkin. And I've got her in, though it ties into something quite interesting. I'll try and remember to come back to if we have time at the end. She was one of the key people in developing X-ray crystallography as a method. Now, one of the things we didn't get into in the experimental design lecture, I thought we might, is how you look at things which are very small. So we didn't get into talking about how you would look at the structure of myoglobin. But in practice, you can't look at things any smaller than the wavelength of the light you are looking at. And that means that you can't get much below bacterial size using optical wavelengths. You can get at much smaller structures using X-rays. And though the data is hard to unravel and you have to use crystals to do so. She got the Nobel Prize for deducing the structure of vitamin B12. But she was also responsible for verifying chain structure of penicillin. Though that didn't come out for a while. Interestingly, a lot of the research being done on this, because it was wartime and because this was important to the wartime effort, was taking place kind of behind closed doors. It wasn't going into the literature because then the Germans would know how to make it as well. Another interesting thing is that Hodgkin is her married name. Still at a time when, as a married woman, she would use her husband's name in her publications. That doesn't tend to happen these days. Her husband, I think, has vanished to posterity, but she has a Nobel Prize. This is an example of an X-ray crystal image of lysozyme, I think. So the problem is to work out how these individual dots can be taken back to give you the atoms in that original structure. And that, I suppose, also brings in, this was Bob's suggestion, is that we might also, while we're talking about milestones, note how important the development of the electron microscope is. Now, the person we would give that as a name, the person who got the Nobel Prize for it, is Rösker, who built it based upon other people's ideas in Germany. Electrons have shorter wavelengths than light. They do have wavelengths, but that means they can see things that are much smaller. Interesting to note in passing that this electron microscope, which was the first, could only produce a magnification of 400 times. Remember that Leeuwenhoek was producing 277 times using single glass lenses in magnifying glasses. But obviously we've gone further since. And that, the importance of this to our understanding of how the cell works can't really be underestimated. This is what you can see with a light microscope. We can do better now because of tweaks to the technology, but this is what's revealed in a similar cell by the electron microscope. And so you can see, this would appear to be a plant cell because we've got chloroplasts in there, we've got the ER, we've got the mitochondria. So we're starting to see what is actually going on inside the cell, the idea that there are organelles and so on. The other thing Bob suggested I know, which I forgot at the beginning, is of course, I've talked about the beginnings of microbiology coming from Leeuwenhoek, but of course it wasn't entirely. There are people going back to the distant past who worked out how to make yogurt, how to make wine, how to make beer. Because these were them working with microbiology, even though they didn't know that that's what they were doing. Arguably in the West, they were critical, because, and you may or may not know, but people in medieval England used to drink gallons of beer a day. It wasn't full alcohol beer, it was called small beer, which only had 1% of alcohol or so in it. But that was critical, because if you drank the water, you died. It was, that may say a lot about the state of medieval Britain and Europe that was the case. But other parts of the world used tea or coffee or whatever, where you're boiling the water, but it was an important element in life in Europe. And I hope I'll be able to get through quickly, because I say we do need to finish on time. So that's one thread coming from cell biology into microbiology, because the other thread coming from those early microscopic images is cell biology in terms of larger organisms. And Joanne was keen that we talk about developmental biology and how that feeds into the history of biology as well. And for a long time, this idea held sway, called preformation. That essentially organisms developed just by getting bigger. And so there's this drawing, actually quite a late drawing, of, this is a sperm cell, and the idea is that you've got a little man inside the sperm cell, and that the new human, or woman, and that the human comes from this just getting bigger. Of course, it's in the sperm cell because women aren't important. Obviously, the important bit has to be coming from man. Attitudes of the time, not mine, honest. And so there is an important change taking place in terms of what's called epigenesis. That the idea that what you have is that organism is coming from something less formed. That you have a process of differentiation that is leading to the complete organism. So the people responsible for this will start with Wolff. And in fact, he did write a book called something like Embryogenesis, but the key one of his text was work on the development of the intestine. You looked at a lot of different species, but particularly in looking at vertebrates, he noticed that you had these layers, what he called leaf-like structures, involved in early development in animal embryos. And that observation is the first coming into the germ layer theory. And was further developed by Panda, though he didn't take it too far forward, because he was essentially only looking at chicken embryos. He had a workable system looking at hen eggs, chicken eggs, and so he verified by close observation that the early stages of embryogenesis in chickens, you have these clear 3 layers. So he's building on these leaves that Wolf had observed, and suggesting that they're important in this particular species. But it's Bayer who verifies that this is what happens in all of the animal species he looked at. Now, he's a very interesting person in terms of biology, because one of the things we're going to slightly divert into is the connection between ontology, so the development of the organism, and evolution. An idea that's called recapitulation. And in his early writings, he seemed to be quite receptive to this idea, though he rejected one of the stronger ideas of it, that you were seeing development from a primitive organism to a complex organism. He was quite clear that that's not what you saw, and his observations are quite correct in it. But what he did pick out that pretty much all vertebrate embryos, chordate embryos, look the same at the beginning. And then soon after that, they start to follow a slightly different path of development, but even at that stage, the mammalian embryos look similar, the bird embryos look similar, the reptile embryos look similar. And it's only as the development proceeds that you see the specific characteristics of each species come through. In his early writings, he was receptive to the idea that this was perhaps evolutionary, but he was connected to some of the at the time, science in Germany was quite strongly influenced by the Romantic artistic movements, and it had this idea that there was kind of a life force or a driving mind behind life. Somehow, living material was different. That's an idea called vitalism. And as a result of that, when Darwin published, even though Darwin was picking up some of his observations, he rejected that mechanistic explanation for evolution very strongly, to the point where he went on to repudiate his earlier thinking on evolution. Person most closely associated with this idea is Ernst Haeckel, who was a strong recapitulationist. He's now believed to be wrong both on recapitulation and evolution, where he was quite Lamarckian, but he came up with this really quite pithy phrase, ontology recapitulates phylogeny. This idea that you would see an organism going through its entire developmental history, entire evolutionary history as it develops. He was a fairly dubious politically as well, more than a little racist. Now, this is a slide that Joanne drafted, and it draws together something quite interesting and important, which is that we have a number of quite separate fields developing independently, and they aren't at this stage talking to one another. And particularly the people working on evolution have missed there are key pieces that the others can supply that, that they aren't finding. And so while, for example, they had the Archaeopteryx, now, Joanne put in now considered not transitional, but a bird. This, in fact, connects more to the fact that the lines, where do you draw the line between a dinosaur and a bird? And in fact, some people would say that all modern birds are actually dinosaurs. But they had this, but what they hadn't picked up was necessarily how much developmental biology could teach them. There is this story that Joanne has put in. I'm not going to have time to talk it through. There is a video of it here. It's a very sad story which turns upon a flawed idea that emperor penguins were transitional between reptiles and birds. It's completely incorrect. And these 3 people went on a catastrophic expedition to recover emperor penguin eggs in order to test it. And then the eggs weren't even looked at. You can see them in the Natural History Museum, but they weren't used to test this theory, which was in any case wrong. I'm not going to have time to go through these slides, but I'd probably recommend that you come back to them, because they pick up a very important developmental thread, which was realizing that there was how inheritance worked. And of course this was a key element that people working on evolution needed, but they didn't have. And so we have a number of workers who are bit by bit putting together the idea that the chromosomes and nuclei are associated with the inherited material, culminating in the idea that firstly, that the germ cells only, the cells carrying out meiosis, are those involved in inheritance, and that the other cells are irrelevant. That then leads us into Mayer. And Mayer we'll be coming back to in the lecture on the modern synthesis. This is the key point at which all of the pieces came together. Mendel, Darwin, subsequently, of course, Watson and Crick. And it's these giving us the mechanism of heredity and the mechanism which explains how evolution works. This is the foundation of modern biology. Okay, thank you very much. Sorry that was a bit rushed.